Welcome back to the deep dive. Today we are tackling one of the most frustrating paradoxes in modern health.
Right.
We're all told that a healthy, plant-rich diet is, universally good for you.
Eat your vegetables.
Exactly, eat your vegetables. But we've all seen it firsthand. Two people can eat the exact same healthy salad, follow the same diet, and get just wildly different results.
And that intense personal variation is exactly what we're digging into. Our mission in this deep dive is to shortcut you right to the cutting edge of nutritional science.
Looking past that simple food pyramid.
Way past it. We're looking into the hidden engine that decides how effective that healthy diet you're eating actually is.
Okay, so let's unpack this. For decades, nutritional research was focused on what, the big three?
Fats, proteins, and carbohydrates. Maybe a few vitamins.
Right, we were looking at maybe 150 nutrients in total. That was kind of the whole picture.
And it was a fundamentally incomplete picture. I mean, what the researchers behind this study found is just astonishing. That our food, especially plant-based food, is so much more complex. They discovered that the human diet contains over 26,000 distinct small molecules.
26,000.
26,000 separate chemical components. And, the vast majority of those come from plants.
Wow. So we went from focusing on 150 things to realizing there are tens of thousands of these signals we're eating. Which ones are we looking at today?
We're focusing on the bioactive ones, the ones that actually interact with our biology. They're called phytonutrients.
Okay.
And the study's ambition was just massive. They systematically mapped 775 of these phytonutrients to the metabolic tools, the enzymes found in our gut microbes.
Using data from thousands of people.
Over 3,000 human microbiomes in a global data set.
And what did that mapping reveal? What's the scale of this work our microbes are doing?
The scale is staggering. They calculated that at roughly 67%.
Two-thirds.
Two-thirds of all microbial enzymes in the human gut are potentially involved in just transforming these phytonutrients. It's not a side job. It's the primary function of the gut ecosystem.
And that is the core finding right there. This means the implication for you, the listener, is that the anti-inflammatory power of that spinach or the heart health boost from that blueberry is decided really by this massive metabolic engine in your lower intestine.
Exactly.
And if you don't have the right microbial crew with the right tools, the health benefits might just be dramatically reduced. The food's effectiveness is totally conditional.
Exactly right. And that brings us to phase one. Understanding how this microbial factory actually works.
Okay.
Our gut microbes have this enormous, diverse enzymatic toolkit that performs what we call biotransformation. And this is so important because often the health benefit doesn't come from the original thing you ate.
So it's not the raw ingredient, it's the product the bacteria make from it.
Precisely. The bacteria are like specialized chefs. They take a compound that might be inert to our own cells and they transform it into a highly active metabolite. A chemical our body can absorb and use to fight inflammation or boost metabolism?
They're turning a raw ingredient into a brand new therapeutic product.
That's a perfect way to put it.
And when you talk about this machinery, what kind of chemicals are we dealing with? Are they simple or...
Oh, they're diverse and complex. The phytonutrients include things like terpenoids, which give plants their smell.
Right.
Flavonoids, which are often pigments in fruits, and alkaloids, like caffeine. The enzymes doing the work are the real workhorses. Oxidoreductases, transferases, hydrolases, they're the essential tools for modifying molecules.
This all sounds a bit like theoretical modeling, though. Mapping genes to chemicals. Did they actually prove this happens in the real world?
They did. They validated their findings using lab assays, proving that common gut species like Eubacterium ramulus have this strong, verified biotransformation activity.
Yeah.
So no, this isn't just theory. These bacteria are actively doing the work.
But this is where the personalization problem really hits home. This potential isn't the same for everyone.
Not even close. It shows extremely high inter-individual variability. They saw huge differences geographically. Across the thousands of people in the study, the number of phytonutrients in individuals' microbiome could even process range from just 264 all the way up to 620.
That's a staggering difference. That means your neighbor, eating the exact same kale salad, might be getting twice the benefit just because of their microbial lottery.
And it's shaped by more than just chance. We see these clear geographical patterns. Microbiomes from people in Africa and Asia were significantly different from others, which probably reflects regional diets that have been around for generations.
And is this potential fixed when you're an adult? or can it change?
It absolutely changes. And often, not for the better. Your own factors matter a lot. They found that the ratio of these beneficial enzymes was negatively correlated with age.
So this metabolic capacity tends to go down as you get older?
It does.
That's a huge implication for aging. The older we get, the less horsepower we have to extract the benefits from our food.
That seems to be the case. And what's more, they saw a loss of these enzymes as body mass index, or BMI, went up.
Okay.
So the metabolic changes that come with gaining weight seem to be tied to losing the ability to process these healthy components.
But the most dramatic proof of this flexibility came from that immigration study, didn't it?
Yes. They looked at Thai immigrants moving to the U.S., and even with only, modest dietary changes, their enzyme composition became significantly more similar to U.S.-born participants over time.
Wow.
It shows your current diet actively shapes and maybe even erodes this potential much faster than we thought. The microbiome adapts to what you feed it today.
And that dramatic shift brings us to phase 2, the core problem of disease. If diet reshapes your microbes, what happens to your ability to process healthy food when your microbes become suboptimal, or what do researchers call it? Dysbiotic.
Dysbiotic, exactly. It just means the gut's bacterial balance is unhealthy. And the study's central hypothesis was that healthy and diseased people might have, vastly different capacities to process healthy foods.
And they found evidence for that.
Overwhelming evidence. The potential to metabolize healthy foods is significantly altered in disease.
Which strongly suggests that once you have that dysbiosis, that suboptimal microbiota, then broad, one-size-fits-all dietary advice might have very limited value.
You might be eating the perfect diet, but your engine is just clogged.
And this is where the power of the data really comes in. They used machine learning models across 3 different diseases.
Right. Inflammatory bowel disease, or IBD.
Which is chronic gut inflammation.
Colorectal cancer, CRC. And non-alcoholic fatty liver disease, NAFLD.
And they could predict disease just based on these enzyme levels.
Precisely. They found that just by using the abundances of these phytonutrient processing enzymes, they could accurately tell the difference between healthy and diseased individuals. The ability to process healthy food was a predictor of health itself.
That's a critical finding. It's not just that sick people have different guts. It's that their guts are chemically compromised in a way that's directly related to their diet. Let's get a concrete example.
Okay, take the IBD model. We know tryptophan, an amino acid you find in healthy foods like garlic, is important.
Right.
In healthy people, they found a specific enzyme from a common bacterium, Elistipes goldii, that was highly predictive of being healthy. This enzyme converts tryptophan into something called indole.
And indole is important because it's a powerful signaling molecule that helps reduce inflammation in the gut lining.
Correct. But here's the problem. They found this crucial indole-producing enzyme was significantly less abundant in IBD patients.
Meaning their ability to create that anti-inflammatory product from a simple, healthy food component is it's compromised.
Severely compromised.
But wait, is the study suggesting the loss of the enzyme causes the disease, or is it a result of the inflammation? Because that's a huge difference for treatment.
That's a great question, and the study can't prove causation. But the correlation suggests this devastating feedback loop.
Okay.
The dysbiosis in the disease might reduce the body's ability to get anti-inflammatory benefits from the diet, which then makes the inflammation worse.
So the IBD patient eats the garlic, but they're getting dramatically less protective power from it than a healthy person.
The diet is compromised by the gut, which is already compromised by the disease. It just compounds the problem. And they saw similar patterns for NAFLD, the fatty liver disease.
Similar link.
A clear association between a specific enzyme and a compound called tranexamic acid, all linked to NAFLD status. The big takeaway is that this altered capacity to biotransform healthy diets may compromise their beneficial effects, and it's probably different for each food and each disease.
So eating well is only half the battle.
The other half is making sure your microbial community is ready to do the work.
Which leads us perfectly into phase 3, the in vivo proof, the real-world test.
Right.
To settle whether these microbes are really essential, they designed a controlled experiment with a known anti-inflammatory food.
The strawberry. They used a mouse model of colitis, which is a common type of intestinal inflammation.
And they compared 2 different groups of mice.
Exactly. The first group were SBF mice-specific, pathogen-free. They had a normal, healthy gut microbiota. Our baseline
And a second group?
were germ-free mice, or GF.
Yeah.
There had absolutely no gut microbiota at all. Zero bacteria.
Let's start with the SBF mice, the ones with the healthy microbial factory. What happened there?
The results were textbook. Supplementing their diet with strawberries significantly improved their overall health.
In what way?
They had less weight loss, a lower disease activity score that measures symptoms like diarrhea and bleeding, and crucially, less actual tissue damage under the microscope.
So the healthy diet worked perfectly because the microbial engine was running.
It was present and functional, yes.
Now, for the germ-free mice, what happens when you feed an anti-inflammatory food to a body that can't do the biotransformation?
Well, in the GF mice, the strawberry diet only partially helped.
Okay, so it wasn't a total failure.
Not a total failure. It did reduce their disease activity score a bit. It delayed some symptoms, improved stool consistency.
Some benefit, but limited.
Very limited.
Yeah.
Because the critical finding came from the histology looking at the tissue itself. That analysis showed no difference in the actual pathological damage compared to the control mice that got no strawberries.
The core inflammation in the tissue was unchanged.
Unchanged.
That is the smoking gun that wraps this whole thing up. The conclusion is just undeniable. The full-spectrum, deep tissue, anti-inflammatory benefit of strawberries is at least partly mediated by the gut microbiota.
The microbes have to be there, and they have to be active to fully process those healthy components into the beneficial products that do the real work.
So connecting this all back, the study is really suggesting that just consuming healthy plants might be insufficient if your microbiome can't process them.
Exactly. We have to fundamentally revisit this idea of a universally healthy diet. It only benefits you if your gut is equipped to use it.
This opens up some incredible avenues for truly personalized nutrition, though. Shifting the focus from just, dieting to optimizing that human-microbe partnership.
Right. Instead of just saying, eat more plants, we can look at combining specific foods with the specific microbes needed to process them.
And they found data supporting that, didn't they?
They did. They found that small groups of gut bacteria, as few as 11 species, could work together to perform complex biotransformations that the commercial, off-the-shelf probiotics simply couldn't do.
They just don't have the genetic potential.
Right.
So that points toward precision tools, maybe next-generation probiotics or even functional foods where the transformation has already happened before you eat it.
Through targeted ex vivo fermentation. Imagine a yogurt where the active anti-inflammatory compounds from berries were already made bioavailable for you.
It completely revolutionizes what we think of as food processing.
It does. And to bring it all back home, linking back to that personal variability, the study showed that negative correlation between age and these phytonutrient-associated enzymes.
That decline is universal as we get older.
It is. So, given this data, what specific fermented foods or maybe even tailored microbial supplements should you consider exploring to make sure your body can still extract the full anti-inflammatory power from your plant-based diet as you get older?
Something to chew on until our next deep dive.